Using synthetic disaccharide derivatives to mimic and dominate the biofunctions of rhamnolipids produced by Pseudomonas aeruginosa, molecules that mimic and dominate toxic molecules produced by bacteria. Why research this? With the Yan-Ying Luke research team in the chemistry department of Syracuse University, specializing in designing molecules that are able to control the biofunctions of bacteria, they could make such groundbreaking discoveries for biology and pharmacology. Their most recent creation is a class of molecules called synthetic disaccharide derivatives, aka DSDs, which take over the chemical signaling of rhamnolipids to control activities such as biofilm formation, bacterial adhesion, and swarming motility. Pause. What's a biofilm? Well, biofilms occur when individual bacteria, in a way not fully understood, organize into a community that behave like a single organism. They can attach themselves to a human intestine or an implant such as heart valves, and then the cells grow and divide, forming a dense mat that's many layers thick. When there's enough bacteria in the developing biofilm, also known as a quorum, the molecules secrete a sugary glue and form a mushroom-type shaped structure. And the glue acts as a protectant, as a resistant force against antibiotics, toxic chemicals, and the body's own immune system. Let's go back in time a little bit. The first semi-successful synthetic disaccharides were published in the Dictionary of Applied Chemistry by Sir Edward Thorpe in Volume 1. The very first disaccharide was lactose, or milk sugar. Lactose is hydrolyzed by mineral acids to dextrose and galactose. It is a galactoside, that is, the active aldehyde group is in the dextrose residue. At the time, it's uncertain whether the primary alcohol group or the secondary alcohol group of the dextrose molecule takes part in the union from the galactose residue. Accordingly, lactose has one of the following formulas. The synthesis of lactose has not been effected, but an isomeride named provisionally isolactose was obtained by Fischer and Armstrong by a synthetical action of kefir lactase on a concentrated mixture of equal parts of galactose and dextrose. It was isolated as a phenolysolactosazone. A galactosidoglucoso was prepared by the same workers from acetylchlorogalactose and shown to be probably identical with melibiose. With that foundation, George O'Toole and his team in 2006 were able to publish their findings on bacterial quorum sensing, signal circuits, and implications for biofilms and disease. An activity reversal was revealed in the early discovery of the signaling molecule isohomoserine lactone that controls the quorum sensing of Photobacterium fischeri. Also, the activity reversal of rhamnolipids was also indicated by a seminal work of O'Toole and co-workers. Yan-Ying Luke's research team has been able to build upon to create their own synthetic disaccharide derivatives that have the potential to be drug molecules. They found that the activities of both the rhamnolipids and DSDs were very concentration dependent. The higher the concentration was, the better the DSDs were able to counteract the activation of swarming motility and biofilm formation of the rhamnolipids. The research team came to notice that even though at higher concentrations the DSDs were better apted to take over the rhamnolipids, they also noted that the concentrations were respectively lower than that of the rhamnolipids needed to activate the swarming and the bacterial adhesion and biofilm formation. They were able to tell this by the tendrils that formed during the experiment. After designing and synthesizing two chemical libraries, a collection of molecules, Luke's third one contained two DSDs whose structures were able to dominate the function of rhamnolipids. This discovery offers the potential for many applications, especially with rhamnolipids being non-toxic, biodegradable, and being able to withstand extreme salinity and activity. Oh, and many have tried and many have failed to warp rhamnolipids into therapeutic agents. Hopefully, Luke will bring us to victory. Keep calm, Luke, and no pressure. Thank you.